Hello, dear colleagues. My name is Maria Vlasenok, and I am a PhD student in Professor Perlushin's lab at Skoltech. Today, I want to tell you about my work on interplay between cleavage and polyadenylation and splicing. It suggests that splicing can counteract premature cleavage and polyadenylation in introns. The majority of transcripts generated by polymerase II are cleaved and polyadenylated. At least half of human genes undergo alternative polyadenylation. As you can see on the scheme, alternative polyadenylation sites can be located in the same terminal exon or upstream of it, in which case they are in introns. About 20% of human genes undergo intronic polyadenylation. However, there are much more cryptic intronic polyadenylation sites, and research suggests that cleavage and polyadenylation machinery recognize these cryptic sites. So why don't we see much more intronic polyadenylation events? We hypothesize that this is because splicing can excise intronic polyadenylation site before cleavage and polyadenylation takes place. Indeed, if the cryptic intronic polyadenylation site is spliced out, it cannot lead to generation of truncated transcript. Our goal was to look for evidence of interplay between splicing and intronic polyadenylation. To do that, we need data that allows to simultaneously assess alternative splicing and EPA. Matched datasets are very limited, because to study splicing we need RNA sequencing dataset, and to study intronic polyadenylation, normally oligo-DT primers-based methods are used, like 3' sequencing, 3' base, polysec, etc. So the easiest way here would be to use poly plus RNA sequencing to study polyadenylation too. Although there are many methods to identify polyadenylation sites from RNA sequencing data, most of them do not work well for intronic polyadenylation sites. So we developed our own pipeline. It's based on reads with adenine-rich soft clipped ends that do not align to the genome. These reads are called poly-A reads. Genomic position of soft clipped start is a candidate polyadenylation site. We pool these polyadenylation sites between samples, and to reduce false positives, delete ones that are located at genomic A runs, and filter polyadenylation sites by poly-A read support and diversity of the poly-A read supporting them. Previous attempts to use such methodology resulted in recall which is lower than 10% due to scarcity of poly-A reads. So we decided to apply this technique to large-scale dataset. We picked the RNA sequencing dataset from Genome Tissue Expression project, and as a result, we got 300,000 polyadenylation sites. To verify our path set, we compared it to poly-A site consolidated atlas, which was gathered from publicly available 3' sequencing datasets by the group of Professor Zavalan. As you can see on the right, precision recall curves for comparison of both these sets to annotated transcript ends from gene code are very similar, which speaks to validity of our path set. Next, we used the obtained set of polyadenylation sites to analyze the distribution of polyadenylation events in the genomic regions. Expectedly, number of paths and path density was highest in 3' UTR regions. However, more than 60,000 unannotated polyadenylation sites were also found in open reading frames. Among them, more than 40,000 were located in introns. Moreover, fraction of poly-A reads, which can be a proxy for frequency of polyadenylation events, was highest in the intronic regions. All this data confirm that intronic polyadenylation is frequent and widespread. Any intronic polyadenylation event is associated with expression of an alternative terminal exon. Thus, intronic polyadenylation events are often classified based on the type of this exon. Skip terminal exons are results of cleavage and polyadenylation happening in a cassette exon, while composite terminal exons appear when cleavage and polyadenylation happens in the retained intron. These two types can be distinguished based on relative coverage of the intron beginning. As shown on the scheme, coverage of the intron beginning is much lower than the exonic one for skip terminal exon, while coverage does not change at exon-intron border in case of composite terminal exons. Moreover, we can distinguish between these two types based on the reads aligned to the splice junctions. Skip terminal exons are characterized by non-canonical exon-exon junctions upstream of the polyadenylation site, while continuous reads that span the donor splice site, denoted as C here, mark composite terminal exon. Also, there are reads that support canonical splicing, which are denoted as A. So using these data, we also can estimate the rate of canonical splicing C, which we expect to be negatively correlated with the intronic polyadenylation site usage. To categorize the intronic polyadenylation sites with signal, we computed coverage fold change at pass, coverages near intron exon border, and PSI for each pass in each tissue using GDX data. Then we selected a set of 7.5 thousand intronic polyadenylation sites expressed in at least one tissues. Express pass is characterized by at least 10-fold coverage drop at polyadenylation site and substantial read coverage in the intron upstream of the site. About 1,500 polyadenylation sites out of these 7.5 thousand can be classified as STE or CTE based only on the annotation. So we analyzed coverages near intron exon border for these annotated sites. Indeed, as expected, the two types of polyadenylation sites show distinct coverage profiles. In skip terminal exon intronic pass, the second window has substantially lower coverage than the first, while for composite terminal exons, the ratio is about 1. The two groups are divided by threshold ratio of about 0.3, as you can see from the plots on the right. Interestingly, coverage profiles for polyadenylation sites that are annotated seem to be the combination of the two groups, which allows to classify these polyadenylation sites into either CTE or STE. Next, we looked at rate of canonical splicing for these sites. As expected, annotated sites are characterized by low rate of canonical splicing. However, surprisingly, other expressed intronic pascs have an additional peak at PSI equal to 1. This peak is incompatible with both CTE and STE models, because expressed polyadenylation sites with PSI equal to 1 imply that canonical splicing coexists with intronic polyadenylation. To look further into this, we examined the coverage profile for these sites. It shows substantial coverage drop at the beginning of the intron, similarly to skip terminal exon pass. So in this case, the intron is spliced out, but part of the intron is not degraded and detectable in RNA sequencing data. At the same time, this part ends with a polyadenylation site. We asked the question, how could these RNA species arise? As you remember, in the beginning we discussed the two canonical fates of an intronic polyadenylation sites. It's either expressed and as a result a truncated transcript is generated, or the polyadenylation site can be degraded as a part of intron ladder. What if it's something in between? If the intron is both spliced and polyadenylated. In other words, our hypothesis is that if cleavage at the polyadenylation sites in the intron starts after the spliceosome has assembled on it, then the second catalytic step of splicing will remove the lariat and cleavage and polyadenylation products, resulting in spliced and polyadenylated intron. We suppose that similarly to any lariat, the spliced polyadenylated introns are linearized in the cell by the debranching enzyme DBR1. Then the two linear products are parts of the intron before and after the intron polyadenylation site. The former segment has a monophosphate at 5' end from the debranching process and the poly A tail that protects it from degradation. A protocol that captures such RNAs has been developed. It's called cleavage. We reanalyzed cleavage dataset and indeed saw enrichment of cleavage coverage at the beginning of introns containing an intronic polyadenylation site. Moreover, the enrichment was more pronounced for intronic polyadenylation sites characterized as spliced polyadenylated intron. Interestingly, we also observe a similar enrichment in dataset from 3' pull-down vitro capping experiments performed by Dr. Malka and colleagues, which essentially capture the same RNA species. These results indicate that SPIs are not 3' ends of distinct polymerase II transcripts initiated and terminated within the intron, but SPIs are a result of interplay between cleavage and polyadenylation and splicing processes. To sum up, we showed that quality of polyadenylation sites identification from polyareads improves substantially with increase in dataset size. We provide a set of 300,000 polyadenylation site clusters from DETECT's data, which can be used to study tissue-specific alternative polyadenylation via other different techniques. Also, we provide a set of around 70,000 intronic polyadenylation sites with computed splicing characteristics that can be used to examine interplay between alternative splicing and intronic polyadenylation. We discussed a model that can be used to distinguish between the two types of alternative terminal exon for these intronic polyadenylation sites. And last but not least, the data suggests existence of prematurely polyadenylated and spliced introns, and this hypothesis is supported by evidence from cleavage and 3' pull-down in vitro capping experiments. This work has been recently published in Genomics and Bioinformatics, and I thank you for your attention and invite you to ask any questions or give me any comments in person, by email, or in the conference web platform. Thank you very much again. Bye.